Hello, everyone. It's Monday, October 26th. How's the connection this morning?
Good.
It's good. You can hear me well? All right. So we are talking about nucleic acids. It's chapter 8. We have 2 lectures on that, Monday, Wednesday. And then next Monday after Halloween, we have our second test. So the second test will be very similar to the first one. So no surprises. And you guys did reasonably well. So I assume that the format was good and I got some positive feedback from many of you. All right. If you do have any more concerns, just let me know. Send me an email. All right. Let me start sharing my screen. Start sharing my screen. And we talked about glycosidic bonds. So we talked about numbering, we talked about the beta configuration of the glycosidic bond, we talked about depurination, right? Which is the removal of the nucleic purine from the ribose unit, very common reaction in DNA damage. All right, so let's talk a little bit about conformational preferences of, let's say, nucleosides. So you can see here, there is a carbon-nitrogen bond at the anomeric position, all right? This is our anomeric position. Oops. Anomeric position. And we have a free rotation about carbon-nitrogen bond. And so you can see the purine, for example, right? It can find itself oriented right above the ribose sugar, or it can be found away from the ribose sugar. Specifically, what we the atoms that we follow for this are, so let's say, so for the purine, it's O4 prime. It's this one, right? This is 4 prime. 4 prime. So this bond, or 4 prime to C1 prime. This is 1 prime. Then we'll go to N9. Go to N9. This is position 9, remember, of a purine. And then C4. If this is 1, 2, 3, 4, it's this bond. So what do we see? So basically, the rotation around this 1 prime and 9 bond gives us the 2 other bonds eclipsing each other. So this will be syn-adenosine, right? From organic chemistry, we know when the 2 bonds are eclipsing each other, they're syn. Now here, we follow the same bonds, 4 prime, 1 prime, 1 prime, N9, and N9, C4, like that. And we can see that this is anti, right? So it's important because different kinds of DNA will, different conditions can actually make this bond rotate inside the DNA, and the DNA can adopt different structures, and RNA as well. So it's important to know where it's coming from, and we'll get to it when we talk about Z form of DNA, for example. So this angle is 0. This angle is 180, right? Anti-conformation. And the anti-conformation is found in normal bDNA. So we'll talk about aDNA, bDNA, and we'll talk about zDNA. So the three main types of DNA that have been identified. And so the normal DNA is referred to as bDNA, and all of these are anti. And so for the pyrimidines, again, we'll start with the O4 prime, O4 prime to C1 prime, like so. C1 prime to N1. Remember, this is 1, 2, 3. Right, you start with this nitrogen, and then you go towards the next nitrogen. And it will be N1 to C2. N1 to C2. So it's 180 degrees. So it's anti. All of these are anti. This is syn. So in pyrimidines, you can see that it's anti because this carbonyl oxygen is important. So if in purines, it's kind of makes sense that anti predominates, right? Because in this form, there's quite a bit of steric hindrance between the purine ring and the ribose sugar. What do you think the problem is with pyrimidines? Why do you think anti is favored? What should we look at? What should tell us that anti is favored? Basically, you look on the left-hand side and on the right-hand side. What do you think?
Would it be because if it was syn cytidine, the oxygens would be repelling each other? Or they can't interact as two negative charges.
Yeah. Well, they don't have negative charges, they have partial negative charges, right? But there's these lone pairs of electrons. Really bad. It will be electron repulsion, right? So the carbonyl from this and the oxygen from the ribose will repel each other because of the lone pairs of electrons. That's right. So, and actually you can see the same kind of, when I said steric hindrance, but you can also see that there's a lone pair of electrons on the nitrogen, right? And there's a lone pair of electrons on the oxygen, which will both repel each other. So same idea. But after all, the steric hindrance is about electron repulsion. When we say steric hindrance, it kind of, I think the first thing that comes to mind for us is kind of no free space, right? Steric hindrance, that means there's just not enough space to accommodate different groups. But in general, it's about bond repelling other bonds, and bonds are composed of electrons, right? So electrons repel each other, and that's where steric hindrance actually comes from. So to say that there is steric hindrance is the same as to say that the lone pairs from the electrons repel each other. It's the same as steric hindrance. So let's clarify that. All right, so there's a tautomerism of nitrogenous bases, also very important characteristic of nitrogenous bases. So as you remember from OChem 2, if you have a ketone, and there is hydrogen at the alpha position, right? The ketone can undergo tautomerization. So this hydrogen can migrate from carbon to the oxygen. And we refer to this, remember the name of this tautomerism? It's right here. Keto. Keto-enol. Well, why is it enol? Is because en of the double bond, right? And ol because of the alcohol. So it's enol form, it's a keto form. Anybody remembers which one is more stable? Nobody? So this equilibrium favors the keto form by far. So the carbonyl group is actually very stable. Carbonyl, very stable. That's something that we always have to remember. Every time you have the possibility to form a carbonyl group, the energy will go down. The energy will go down, and probably some of the most stable molecules are what? For example, carbon dioxide, right? Carbon dioxide has 2 carbonyl groups. Stable. Released in the atmosphere. It's released in the atmosphere and does what to the atmosphere? Why don't we like carbon dioxide to be released in the atmosphere? So it's stable, it'll persist in the atmosphere and will do what? When we talk about pollution, when we talk about when people burn rainforest to cook in their oven, right? So why don't we like that? Why don't we like the release of carbon dioxide? Come on, guys, this is general chemistry. Carbon dioxide is a greenhouse gas. Anybody knows what a greenhouse gas is?
It will like increase global warming, or contributes to it, because it'll like reflect heat like back onto the Earth's surface rather than letting it escape, I guess.
That's correct. Mm-hmm. Why do I have this stuck? Anyway, so yeah, you can write this down. For some reason, my pen doesn't work. Well, it does actually. So it's a greenhouse. Greenhouse gas. It will absorb ultraviolet radiation from the, infrared radiation from the sun, right, and capture that in terms of heat, and dissipate that heat, and contribute to global warming. And the reason for that is because of the stability of the carbonyl. See how far we went from the keto enol tautomerization. We started talking about global warming. Yeah, we can do these kind of connections all day long. That's the beauty of science. All right. So with nitrogenous bases, we have for example, so this is a uracil, right? So you can have the form of the lactam, where you can see that there's a lactam, remember, is a amide, which is a cyclic amide, right? So there's a lactam form. You have one amide, this amide, and this amide. So it's a bis-lactam form. Now, if this amide, if this nitrogen, if this oxygen uses electrons to grab the proton, the electrons from the nitrogen move towards the carbon, and electrons from the oxygen become the lone pair on the oxygen. Then you form the lactim. Lactim is the mono-lactam, mono-enol form. Now, if you do the same thing with the other one, like so, like that, and like that, then you get double lactim. Now, again, the preference is for the lactam form, but there's quite a bit of the aromatic character. right? So you can see these are equilibrium signs, right? Which means that under normal conditions, all three of these are present. They may not be present in equivalent amounts, but all three of them are present. So as we will see, so these kind of tautomeric relationships were crucial in deciphering the DNA double helix structure, because it was not clear for scientists, Watson and Crick, it was not clear which tautomeric forms the nitrogenous bases adopted, right, before they could actually come up with a reasonable structure. Right, but because of the aromatic character, right, so all nitrogenous bases are capable of aromatic systems. They will absorb light in the UV region. So 250 to 170 nanometers is due to the pi star electronic transitions. So all aromatic structures absorb light, UV light in particular. And the reason for that is obviously highly conjugated system of pi electrons. So for example, in benzene, you have these p orbitals, and there is very nice conjugation, right? So it's a system of pi electrons. If they will absorb light, the electrons get promoted to the antibonding pi star molecular orbital, right? And in a way, this is a this is a way for DNA to actually protect itself from ultraviolet radiation. Okay, because so once this is promoted and the amount of energy which is required to go from pi to pi star is small, right? So not sufficient to break any bonds. And then it undergoes what's known as radiationless transitions, right? So mostly probably thermal. So right, this will move a little bit more, and then the energy will be released as heat without damaging any of the bonds. So, so this will protect DNA from the harmful UV light. So it doesn't protect it. It's not 100% guarantee, right? So we still all have to wear sunscreen. We have to wear sunscreen and to protect ourselves from the harmful UV radiation. But if it wasn't for this absorption by the nucleic bases, our DNA would undergo DNA mutations all the time from exposure to harmful radiation. Okay, makes sense? So the aromatic character of the nucleic bases is crucial in DNA function. And this is just the some curves showing that different nucleotides absorb with different extinction coefficients and a different wavelength maxima, right? So remember, extinction coefficient really tells you how much light is actually absorbed, intensity of the absorption. And so adenosine monophosphate, so this is the nucleotide, absorbs light, absorbs much more light than GMP, UMP, DTMP, or CMP. Okay, so based on this, just by looking at this, so tell me, which nucleic bases absorb light better, purines or pyrimidines?
Would it be purines because the hump is bigger, I guess, on the graph?
Yeah, and also by just looking at these numbers. So what's adenosine monophosphate? Is it purine or pyrimidine?
Purine.
And what's guanosine monophosphate?
Same thing, purine.
Okay, so so we have two purines absorbing much more strongly than the pyrimidines, right? And so, so experimentally can we come up with a theory that explains that? So why purine bases absorb light better?
It's because they have 2 rings instead of 1.
And yes, correct. So they have 2 rings, and what does that do? Why the two rings are better than 1?
More aromatic.
More aromatic, yeah. So
Characters?
basically, yeah. So remember when we talk about pi to pi star, it's all about electron conjugation, right? So the more highly conjugated system you have, the better the absorption by light will be, and also the absorption will move towards the visible region. So, for example, you know that highly conjugated systems absorb light very strongly. For example, retinol. Retinol, as you know, it's a vitamin A derivative, right? Why do our parents tell us to eat carrots, or our grandparents more likely tell us to eat parents if you want to see better? If you want to have a good vision, eat more carrots. Have you heard that? No, you haven't.
Yes. Isn't it like the beta carotene in it?
Well, the beta carotene is responsible for the color, right? So the more, and then it's the same idea, that the more conjugated system, the better the absorption of light. So retinol is actually it's a different type of vitamin, right? And that's the one actually that is used in the retina, right? Basically in the eye tissue that is responsible for the sensitivity to light, right? And so this is also a highly conjugated system which absorbs light very well. All right. Now there are minor nucleosides in DNA, something useful to remember. So DNA can be methylated. This is one of the main modifications of DNA, which is done after DNA synthesis. And methylcytosine is the most common type of modification in eukaryotes, and is found in bacteria as well. And so this is methylcytidine when it's connected to ribose. And so so you can see here, so cytidine normally does not have this methyl group, right? So thymine has methyl group, but here we have methyl group attached to cytidine, which is a rare nucleic base, but it primarily occurs in the promoter regions of various genes. So, and it serves the function of epigenetic silencing. What is epigenetics? Anybody knows from previous classes?
Doesn't that mean that it is a heritable, like it does get passed on, but it's not like the DNA code, it's like something that's still passed on though, but it, like the methylation. So it has to do with the, like turning on and turning off of genes, is that right?
Yeah. So with genetics, with the genetic system, there's nothing you can't really do anything about it. You're born with a specific genetic makeup, right? All cells on your body from all tissues will have exactly the same genes, and there's not much you can do. There's something called gene therapy, which is a still developing area. It's very hard to deliver pieces of DNA into cells. But it's been developing for many years, and we still don't have a good system for that. But for epigenetics, so we can actually not control, but we can influence which genes are transcribed and which are not, right? Or transcribed more, transcribed less. So people talk about epigenetics in terms of, the environmental stimuli, what you eat, what you do, right? You can, so that's all epigenetics. You can more or less control which genes are produced more, which genes are produced less. And for example, cancer cells, they love to play with genes. They love to do epigenetic stuff by silencing genes, for example, of tumor suppressors, right? So the proteins which will prevent them from growing. So they're actually suppressed in cancer cells. And one of the ways to suppress tumor transcription is, the protein transcription is by methylating the promoter region of various genes. And so methylation has a crucial role in that. So that's a minor base, but a very important one. Now, other methylations also play a role in eukaryotes and prokaryotes. Hydroxymethyl at 5 position, apparently it sort of plays a role of methyl group, but it's much more rare. But as long as you remember methylation and what it does, that's all I need to know. That's all you need to know from here. And so, yeah, so way to mark DNA of the cells. And also another function of that is you can mark your own DNA so that if you employ DNases, which are enzymes that degrade DNA, if your immune system employs DNases to degrade foreign DNA, or primarily in prokaryotes, I guess, then they can protect their own DNA by methylating that and key positions. And then the DNases, when they recognize the methylation, they can tell apart whether it's host DNA or whether it's the DNA of an invading organism. Okay. Now, there are also minor nucleosides in RNA. So the two important ones are here, inosine and pseudouridine. So inosine is shown on the next page, and you can see here. So this nucleic base is actually, when it's not attached to ribose, is known as hypoxanthine. Hypoxanthine. Okay. And so hypoxanthine is actually a deaminated version of adenine. So remember, adenine will look like this. NH2. Like so. Like so. So in adenosine, it will be there will be a ribose here. Ribose. So with the ribose, this will be adenosine. And so inosine comes from adenosine through the loss of this NH2 group. Right, so if you lose this NH2 group, you go there. And inosine is an important nucleoside in RNA. Specifically, it occurs in the wobble positions of anticodon tRNA, right? So remember, the messenger RNA brings the code, codes for for amino acids, and tRNA has an anticodon system that recognizes the codon system. And so the first two letters in the codon are constant, but the third one can be wobbly, can actually change, and that actually enriches the genetic code, something that you actually learn in biology. So we're not going to go into detail of recognition of the genetic code and the protein synthesis, but it's important to realize that so the inosine plays a big role in protein translation, right? So when the tRNA brings the amino acid to the ribosome. Now the second one is pseudouridine. Pseudouridine. Now this found widely in tRNA and R, ribosomal RNA. So you can see here, normally in the uridine, this is carbon 1, right? This is carbon 1. And so the nitrogen 1, and the ribose will be linked to this nitrogen at this position. But there are enzymes that can actually isomerize a uridine, and actually instead of ribose being attached here, they will move it over there from 1 to 5. What do you think? What do you think that accomplishes? So obviously, if you look at these two, and the only difference here, it's attached to carbon, and here it's attached to the nitrogen, but the rest of the molecule is going to be the same for both, right? What do you think is accomplished by this isomerization?
Does it make like a kink in the DNA, or in the, I think you said it was RNA that it's found in?
It's RNA. Yeah, we're talking about RNA right now. This?
I don't know, my only guess would be that it makes like a kink so that it might be easier to I don't know if it has something to do with how it's transcribed.
This is just kind of like a guess, but when you move that ribose over to the carbon, does that like free up that nitrogen for like some chemistry to interact there?
Possible. We don't know about that. Well, maybe there are papers that talk about that. I haven't seen, but it's not in a textbook clearly. But one thing that comes to mind, at least to my mind first, is the stability of the anomeric bond. So now imagine if you have you have ribose, right? Have a ribose. Now you have a nitrogen here, like so, versus you have a ribose. You have a ribose, and now you have a carbon, right? So if you talk about the formation of the A basic sites, remember I told you about A basic sites when you have a deep urination or deep pyrimidination, I guess. Never heard that, but why not? So when this bond breaks and this bond breaks, so which bond is more stable, carbon-nitrogen or carbon-carbon? Can you break carbon-carbon bond easily? Remember the way it's going to happen? We talked about that last time. You're going to have to protonate this, right? You're going to have to protonate. You told me yourself that purines are going to be protonated more easily than pyrimidines, but pyrimidines will be protonated as well. This will come in and it will kick out this nitrogen, right? Remember that? And now let's try to do the same thing. We're gonna protonate this ring, pyrimidine ring. If this can come in and kick out this carbon. So which process is more favorable? The top one or the bottom one? Think of electronegativity of nitrogen versus carbon.
Wouldn't the top one be more favorable?
And what's the explanation?
There's more of a difference in electronegativity between the carbon and the nitrogen versus there's no difference with carbon to carbon.
Right. And so that makes the nitrogen a better leaving group, right? I think that's the point. So obviously, if nitrogen is more electronegative, it can hold the negative charge better and can be protonated more easily too, right? And so it will leave more easily than, well, this will not leave at all. So once this carbon-carbon bond is formed, there's nothing there at all that will break it. Breaking carbon-carbon bond is the most difficult thing in organic chemistry or biological system. So one main function of that would be to make RNA more stable. So for example, if there are, let's say, does it occur in messenger and tRNA both? Let's see. Pseudouridine found, yes, in tRNA, no, in tRNA and ribosomal RNA. So if you have, I guess, sites in these RNAs which are susceptible to the destruction, right? So if the anomeric carbon breaks down to release the base, nitrogenous base, then the pyrimidine can undergo this kind of isomerization to form a stable carbon-carbon bond. And I guess that arises from natural selection, right? So the nature has taken care of the lack of stability of these sites in tRNA and rRNA by summarizing the pyrimidine and making pseudouridine from uridine. And pseudo, remember this prefix usually means it's almost or like so basically like uridine or almost like uridine. All right, so this happens after RNA synthesis, right? So it's a post-transcriptional modification. May stabilize the structure of tRNA. Also says here may help in folding. Maybe what was said earlier regarding the NH. Right, so here there's an H, here there's no, so there's hydrogen bonding possibility here, and there's no hydrogen bonding possibility on carbon 5. So maybe folding becomes important. If you add an extra hydrogen bond, right, then the folding preferences will change as well. So that could be another reason. Yes, that's correct. Good answer. Now, there are a couple others which we're not going to talk in detail about, but you can put sulfur here. So this will be thiouridine. This will also change the preferences, various preferences. Obviously, you change the electronegativity, you change nucleophilicity. You can methylate the N7 nitrogen in guanosine, for example. And by doing so, you can introduce a positive charge, right? So the enzymes that incorporate methyl groups are known as methyltransferases. Very large family of enzymes, methyltransferases. Because methylation is a very common process in biological systems. All right, any questions? So these are minor nucleotides. Okay, and so now we have discussed the monomeric species, right? Nucleosides, nucleotides, nucleic bases, ribose, phosphates. So now it's time to move on to polymeric structures. Guys, could you just hold on a second? Just hold on a second. I need... Okay, I'm back. All right, so now we're going to talk about polynucleotides. So these are polymeric structures. And so they're going to be linked through phosphodiester linkages. Phosphodiester linkages, so let's look at the fragment of DNA, right? So this is DNA. You can see there is no 2' hydroxy. This is RNA, there is 2' hydroxy. Two prime, and there is nothing here at 2'. So that's the main difference between DNA and RNA. And so, so this linkage here, so we're going to link the ribose units through phosphodiester linkage. So remember, so why they call phosphodiester? So remember phosphoric acid. Phosphoric acid. So phosphoric acid can form esters, just like any acid can form esters. And the esters, now there are 3 hydroxy groups, right? So you can form monoester, diester, or triester. So for the structure of DNA or RNA, it's a diester, which is important, right? So we're going to convert 2 hydroxy groups into 2 ester residues and leave one alone. Leave one alone. And so what do we see here? So this is the component from the phosphoric acid. And you can see here, this is one ester from coming from the 5' position of this ribose. And this is the other ester coming from the 3' position of the other ribose, right? So the phosphodiester linkage links the 3' of one ribose to 5' of the next ribose. And so the nucleotides are usually read from the 5' end to the 3' end, yeah? 5' to 3'. So for example, this one will be ATG and not GTA. So 5', A, T, G, 3'. Okay. And for the RNA, can you name the RNA, this fragment of RNA for me?
UGC?
Yeah, UGC. So start with 5' end, go to 3' end, and so this will be UGC. And so when I wrote this 5' and 3' here, but normally you don't. So you just say my piece of RNA is UGC. Okay. Let's see what else is on the slide. Okay, so DNA backbone is fairly stable, and the hydrolysis is accelerated by enzymes, DNases. DNases are important. So, for example, when you, well, there are many of them are actually processing enzymes, right? So, for example, when you consume various types of food, right? So you consume the meat, vegetables, anything that has DNA in it, right? Anything that has cells and nuclei will have DNA in it. And so DNases will come in and chop up those fragments of DNA into constituent nucleotides, which will be then used to, for example, to form host DNA, right? Can be recycled. Or can be, if there's too much of it, it will be broken down, for example, to obtain nitrogen, for example. Yeah. So DNAs are important. Also, they cleave host DNA, also very important process. For example, in DNA replication, right? So when the DNA is replicated, you need to be able to separate the two strands, the two complementary strands, and you will need a piece of DNA to cut the DNA into parts, and DNases, the enzymes will help with that. So but DNA in general is quite stable, and there is a reason, and that's the main reason why natural selection selected DNA as the primary storage molecule. So RNA is unstable, right? So the RNA, you have to continuously synthesize RNA and degrade it, synthesize and degrade. For example, messenger RNA, right? You have to make it from the DNA template. It has to go to the ribosome, has to provide the template for the synthesis of proteins. Then once enough of the protein is made, this messenger RNA has to be degraded, and it will be degraded by a family of enzymes known as RNases. And in general, when you see ACE on the end, that means as an enzyme that degrades something, right? So for example, esterase breaks down esters, esterase. Amidase breaks down amides, right? Hydrolase breaks down things by hydrolysis, through hydrolysis. All right, so but in general, an RNA lasts for a few years in water, but in cells, it's very quickly degraded, just in a few hours, due to these enzymes, RNases. All right, so let's look at the okay, what else do we have? Directionality, 5', different from 3', and we read the sequence from 5' to 3' end. So we just talked about that. And so RNA in general, it may be stable in water, but it's very unstable under alkaline conditions. So you can actually use just a hydroxide ion to break down RNA, or you can hydrolyze using the enzymes, RNases. So RNase enzymes are abundant around us. So for example, RNase P is a ribozyme. So ribozyme is an enzyme which is made primarily of RNA, right? So we know the enzymes which are made of proteins. So ribozymes are enzymes made of RNA. We have in this class, we don't really look at those in great detail, but there are quite a few of them known, right? And so this enzyme processes tRNA precursors. Now there's an enzyme, there's an dicer, which is an enzyme that cleaves double-stranded RNA into oligonucleotides. So which is used in RNAs, in RNA interference technology. So you probably heard about gene regulation, which is a through the types of RNAs known as microRNA. Have you heard of those? MicroRNA or siRNA, known as short interfering RNAs. Have you heard of these short RNA segments which control gene expression? So in science, these are very, there's a lot of research. It's a very recent research. People have identified these very recently. And for example, in our department, chemistry and biochemistry, Professor Du, she studies microRNA as a therapeutic, as therapeutics to treat neuroblastoma cancer in children. So basically to suppress the production of proteins which contribute to tumor development. So she uses microRNAs. And so these microRNAs basically come from a longer piece of RNA. It's a double-stranded piece of RNA, which is chopped up into smaller pieces by an enzyme known as dicer and used in RNA interference technology. All right. Are there any questions? So the big question for comment that results from this discussion is, why is RNA unstable and why is DNA stable? So the main difference between RNA and DNA is obviously the presence of this 2' hydroxyl, right? 2' hydroxyl. So if you want to break down DNA, in DNA, let's say under basic conditions, so you will have phosphodiester bond, (3' going to 5', like so. So in DNA, this hydroxide would have to attack the phosphorus, right? So this will be an intermolecular reaction, intermolecular hydrolysis. And what do we know about intermolecular reactions? Are they fast or slow? So which ones are faster, intermolecular or intramolecular?
Would intra be faster?
Intra will be faster because for the intermolecular reactions, so these two species have to find each other in solution, right? So the OH, kind of swims around, and it'll take a while before it finds this phosphodiester bond. So this will be slow. slow. And with the, so this will be DNA. And the reason for that is because there is no hydroxyl. There's a hydrogen here. Okay. Now with the RNA, on the other hand, so this reaction, the first reaction is intermolecular, but it's a proton transfer. Proton transfer is always a very fast reaction, right? Proton transfer is very fast, but the actual hydrolysis, the actual breakdown of the diester bond is intramolecular, right? So once you remove this proton and release these electrons, the electrons will attack the phosphorus, will kick out this ester, and will lead to the formation of what's known as a cyclic phosphate. So the bottom part where the 5' group is, right? The 5' portion of the RNA will be released and will come off. So this is intramolecular. Intramolecular. Fast. And then you form a cyclic phosphate, and this cyclic phosphate, in the absence of enzymes, water will simply break it down into 2' and 3' monophosphates. It will open the ring and will give you the phosphate here or the phosphate there. In the presence of an enzyme, it will primarily give you obviously 3' phosphate. So the fundamental, you can see there's a very beautiful chemical mechanism which explains why RNA is less stable and why DNA was selected to store the genetic information. You see what it comes down to? It comes down to a simple concept in organic chemistry known as enchimeric assistance. Anybody remembers what that means? Also known as neighboring group participation. Enchimeric assistance, basically the assistance of this hydroxy group, right? No hydroxy group, no hydrolysis. There is a hydroxy group, the reaction becomes intramolecular, fast, and the phosphodiester bond breaks down quickly. Right, so enchimeric assistance from organic chemistry 2 is responsible for the natural selection of DNA as this storage information. Right. Any questions? Okay. All right, so as far as the hydrogen bonding, so we will just look in a moment at the structure of double-stranded DNA, which was deciphered by Watson and Crick. And so what's important there is that the two bases can hydrogen bond to form a base pair, right? And I told you that the important thing is the selection of the right tautomers. So if you just have a polynucleotide, so if you have just try to link two bases together, there are lots of base pairs are possible. But in the polynucleotide, once you link them through the ribose backbone, only a few possibilities exist. And always keep in mind that A pairs with T and C pairs with G. So these are, purine always with pyrimidine, okay? And obviously in RNA, so this will be in DNA, in RNA, it will be A pairs with what?
Uracil.
Uracil. A pairs with U. And C still G. C still pairs with G. And so here's what the double stranded DNA looks like, right? So you can see here, so there's a phosphate backbone, right? So through the phosphate link through the phosphodiester linkages. And then we see G and C. So G and C, there are 3 hydrogen bonds, and A and T, there are 2 hydrogen bonds. So let's look at the 2 hydrogen bonds. So adenine, so on this side, and thymine on this side. So here, remember in adenine, so I told you that this hydrogen is not attached to a heteroatom, it's attached to carbon. And for that reason, there is no hydrogen bond here, right? No hydrogen bond here. And so there are only two possibilities. This is the hydrogen bond donor, and this is hydrogen bond acceptor. So they switch their roles. And 2 bonds are possible, about 3 angstroms long. Now here with G and C, there are 3 hydrogen bonding possibilities. So the hydrogen bond acceptor, donor, 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 acceptor, acceptor. So switch roles. Okay, so this is something that you should be able to recognize and explain why G pairs with C and A pairs with T, right? And obviously C1 prime, that's the ribose, that's the anomeric position. All right, as far as the structure of DNA. So there was a lot of research, a lot of historical discoveries that eventually led to the discovery of the double helix. So most of the developments took place between the end of the 19th century and the beginning and the first half of the 20th century. So originally, a scientist, Mischler, isolated a substance known as nuclein from cell nuclei, and showed that it was the substance responsible for the storage of the genetic information. So for a long time, scientists believed that it was proteins which are responsible for the storage of genetic information. And the main reason is that obviously that proteins have 20 amino acids, nucleic acids have only 4 nucleotides. Then intuitively, right? So you would, when somebody says there's 20 here and there's 4 there, which is more capable of storing the genetic information? Which one has more diversity, right? And so scientists obviously favored proteins over that. And so Mischler was the discovery was revolutionary in a way that it was shown that the DNA actually possessed the capability to store the genetic information. Then when the nuclein was hydrolyzed, it was found that it consisted of these 3 fragments, consisted of phosphate, consisted of nitrogenous base, and a pentose. So which kind of sugar it was not known yet, but it was known that it was a pentose. And so eventually, in the beginning of the 20th century, the structure of DNA was deciphered. Chemical analysis showed that the ribose units are connected to each other through phosphodiester linkages, and that the pentose is a ribofuranose or ribofuranacide, if you referring to that as the nucleoside. Okay, so these were all the discoveries that originally led to the structure of DNA. But then the question is, how does DNA actually, how is it packed? How does it orient itself in 3-dimensional view? So here are the experiments by Franklin and Wilkins. paved the way for the final discovery. So these two scientists, they were able to obtain an X-ray structure of a DNA fiber, shown here. Now, it doesn't tell us very much because there are specific signs that crystallographers or X-ray people look for. So for example, the cross means helix, right? So these, the dark black areas, so these indicate the repetitive nucleic bases, right? So if you have the same nucleic basis over and over, then the X-ray will deflect more easily when there's the same material, right? The same kind of environment. So and it will deflect in the same area. So eventually, based on all these pieces of data, and another important one was by the scientist named Shargaf. Shargaf, who basically said, or who basically hydrolyzed the DNA and found that the number of G's I should write this down like this. Number of G's equals the number of C's. And the number of A's equals the number of T's. And so from that, it was clear that A pairs with T and G pairs with Z with C. And double helix was the discovery which fit all the experimental data. And so Watson, Crick, and Wilkins shared the Nobel Prize, and Franklin was not given a Nobel Prize. She actually died soon thereafter in 1958. So if the DNA was discovered in 1953, or the double helix in 1953, so she died 5 years later. So, so there's a lot of actually controversy. Obviously, the Nobel Prizes are not given they're only given to living people, right? They're not given what's the word, posthumously? How do you pronounce that? Do you know the word I'm talking about?
Like postmortem, is that the word, I guess?
Yeah, but there's a word like posthumously or something like that. Yeah. So obviously there's a lot of controversy there that some people think that the Nobel Prize should have been given earlier when Franklin was still alive, but because she was a woman, she didn't get one. So there's there's a lot of historical books written on the subject. So if you're interested, you can read about those. And so these are the heroes: Watson and Crick, Franklin and Wilkins. And so here's what they discovered. Basically, the DNA is a double helix. So the ribbon here is the phosphodiester backbone, right? So the ribbon is fully negatively charged. Remember, this phosphate will have a negative charge, right? So the whole bunch of negative charges on the outside of DNA. So DNA is negatively charged. DNA is negatively charged everywhere. And then the, so this will be phosphoryl backbone. And the nucleic bases will actually protrude inside of the double helix and form the hydrogen bonds, form base pairs with each other, right? If there's guanine on one side, so if there's guanine on one side, there will be cytosine on the other side. If there's thymine on one side, there will be adenine on the other side. So, and also base pairs stack. So you can see they're kind of parallel, they're like steps of a ladder. There's a very known phenomenon known as stacking of aromatic structures. So for example, benzene in solution can do the same thing. It's like two molecules of benzene. They are parallel to each other and they're kind of attracted to each other like by a glue. What this allows one to do is this allows one to reduce entropy as well through the hydrophobic effect. Remember, for example, benzene, if you put this in water, right, it hates that. It will not dissolve. The water molecules will have to organize themselves around benzene. Entropy goes up. But if the two molecules of benzene find each other in water, they will stack and they will squeeze the water out of there, right? So it's called base stacking. Or this one is base, it's just aromatic structure stacking. Stacking. So it's a phenomenon well known in organic chemistry, and nature does the same thing. Or actually we do the same thing as nature used to do for many years, many millions of years. So so this and so the bases will be inside, now drawn to each other through hydrogen bonds and through stacking. So that keeps the two strands together very tightly. And the third important observation here is that there is what's known as a major groove and there is minor groove. So that's a consequence of the double helical structure. And major groove is primarily where the proteins bind. So for example, proteins which recognize DNA and perform functions, for example, DNA polymerase. DNA polymerase, anybody knows what DNA polymerase does?
It'll like help synthesize the complementary strand, I think, in transcription.
Replication. So transcription will be synthesis of RNA, right?
Yeah, I got them confused.
Yeah, so that will be RNA polymerase, right? So RNA polymerase will make the messenger RNA from DNA template. But if you want to synthesize the complementary strands, basically if the cell undergoes cell division, right? So you want to double the amount of DNA present in a cell by synthesizing daughter strands, then then the DNA polymerase is utilized. And so the enzyme, so you mentioned RNA polymerase, that's good. So RNA polymerase or DNA polymerase, all these enzymes will bind to the DNA into this major groove. There's more space, right? And so large proteins can find more room in a major groove. Now, drugs, for example, anti-cancer agents, which damage DNA of cancer cells, right? There are a lot of chemotherapy agents on the market, which bind to DNA and introduce DNA damage in cancer cells. They will bind to minor groove. So small molecules prefer the minor groove, large molecules prefer the major groove. And so here are some dimensions. So there's, for example, the distance between the stacked base pairs is 3.4 angstroms. And there's in bDNA, the actual, it's called the helical turn, one helical turn. I think it's called a pitch. I'm not correct, if I'm correct. Anybody knows pitch, helical pitch? I'll write this down and I'll check later. I'll let you know if this isn't correct. Is 36 angstroms. So it's almost like 10 base pairs, but 2 angstroms a little more. So basically one helical turn is actually in normal DNA is 10.5 base pairs. So these are two different other kinds of representations of DNA. So these are from basically use sticks as the bonds, right? Also, you can see clearly the base pair, this base pairing, stacking, and the phosphoryl backbone, right? All these 5-member trina riboses, right? And all the, so this is a phosphorus of the phosphodiester link between the 2 riboses. And then there's a space filling representation. Also very clearly, you can see the major groove, and the minor groove. So we'll stop here. We still have one more lecture. So we'll talk a little more about nucleic acids. We'll talk more about RNA, DNA, some 3-dimensional structures. We'll talk about polymerase chain reaction a bit. And then we'll have our test next Monday. Any questions, guys, about anything that we've talked about today? No? All right, then I'll see you on Wednesday. Email me if you have any questions and throw some questions into the discussion forum. Bye, guys.
Bye, thank you.
Thank you.